Let's talk about the cell organelles. As we mentioned before, they are the working unit of a cell. Each organelle performs its own set of activities and functions. Let's look at them one by one, starting with the nucleus. The nucleus is the most prominent and the largest cellular organelle, occupying 10% of the total cellular space. It plays an important role in cell division as all the genetic material is stored in it. Most of the cells have only one nucleus called uninucleated cells. Few types of cells, like skeletal muscle cells, have many nuclei called multinucleated cells. Structurally, a nucleus is comprised of a nuclear membrane, nucleoplasm, chromatin, chromosomes, and nucleolus. The nucleus serves as a brain of a cell. Its functions are control of all cellular activities, control of cell division through genes, synthesis of RNA, sending genetic signals to cytoplasm via mRNA, formation of subunits of ribosomes, storage of hereditary information. The endoplasmic reticulum is a network of tubular and microsomal vesicular structures which are interconnected with one another. It is a membrane-bound organelle. It serves as a connection between the nuclear membrane and the cell membrane. The two types, rough and smooth ER, are interconnected and continuous with one another. The rough ER is called so because of its rough, bumpy appearance that comes due to the attachment of granular ribosomes to the outer surface. The function of rough ER is to synthesize protein and degrade worn-out organelles. Smooth endoplasmic reticulum. It is the endoplasmic reticulum with a smooth appearance. It is also called the agranular reticulum. It is formed by many interconnected tubules. So it is also called the tubular endoplasmic reticulum. Its functions include the synthesis of lipids and steroids, role in cellular metabolism, storage and metabolism of calcium, catabolism and detoxification of toxic substances. Another important cell organelles is Golgi apparatus or Golgi body or Golgi complex is a membrane-bound organelle involved in the processing of proteins. It is present in all the cells except red blood cells. It consists of 5 to 8 flattened membranous sacs called the cisternae. It has two ends or faces, namely cis face and trans face. Cis face is positioned near the endoplasmic reticulum. Reticular vesicles from the endoplasmic reticulum enter the Golgi apparatus through the cis face. The trans face is situated near the cell membrane. The processed substances make their exit from the Golgi apparatus through the trans face. Major functions of the Golgi apparatus are processing, packing, labeling, and delivery of proteins and other molecules like lipids to different parts of the cell. Because of this, the Golgi apparatus is called the post office or the shipping department of the cell. Let us talk about its functions a bit more. It has 3 main functions. 1. Processing of materials. Vesicles containing glycoproteins and lipids are transported into the Golgi apparatus. Here, the glycoproteins and lipids are modified and processed. 2. Packaging of materials. All the processed materials are packed in the form of secretory granules, secretory vesicles, and lysosomes, which are transported either out of the cell or to another part of the cell. 3. Labeling and delivery of materials. Finally, the Golgi apparatus sorts out the processed and packed materials and labels them, depending upon the chemical content for delivery to their proper destinations. One of the vesicles that buds off the Golgi complex is known as the lysosome. Lysosomes are the membrane-bound vesicular organelles found throughout the cytoplasm. The lysosomes are formed by the Golgi apparatus. The enzymes synthesized in the rough endoplasmic reticulum are processed and packed in the form of small vesicles in the Golgi apparatus. Then, these vesicles are pinched off from the Golgi apparatus and become the lysosomes. It has many small granules which contain hydrolytic enzymes. you should know there are two types of lysosomes. Primary lysosome, which is pinched off from Golgi apparatus. It is inactive despite having hydrolytic enzymes. And secondary lysosome, which is the active lysosome. It is formed by the fusion of a primary lysosome with a phagosome or endosome. Lysosomes are often called the garbage system of the cell because of their degradation activity. About 50 different hydrolytic enzymes, known as acid hydroxylases, are present in the lysosomes, through which lysosomes execute their functions. Its functional activities include degradation of macromolecules, degradation of worn-out organelles, removal of excess secretory products, secretion of perforin, granzymes, melanin, and serotonin. A cell needs energy to perform its activities. This energy is produced by a very important organelle known as mitochondria. The mitochondrion is a membrane-bound cytoplasmic organelle concerned with the production of energy. It is also called the powerhouse of the cell. It has a double membrane structure. The outer membrane is smooth and encloses the contents of the mitochondrion. This membrane contains various enzymes such as acetyl coenzyme, a synthetase, and glycerol phosphate acetyltransferase. The inner membrane is folded in the form of shelf-like inward projections called cristae, and it covers the inner matrix space. Cristae contain many enzymes and other protein molecules which are involved in the respiration and synthesis of adenosine triphosphate. ATP cristae are collectively known as respiratory chain because energy-building process such as Krebs cycle, electron transport chain, and beta-oxidation of fatty acids occur in the matrix. Its functions include production of energy, synthesis of ATP, initiation of apoptosis. Apoptosis is defined as the natural or programmed death of the cell under genetic control. This type of programmed cell death is a normal phenomenon, and it is essential for normal development of the body. Some smaller but essential organelles include centrosomes and microtubules. The centrosome is the membrane-bound cellular organelle situated almost in the center of the cell, close to the nucleus. It consists of two cylindrical structures called centrioles, which are made up of proteins. Centrioles are responsible for the movement of chromosomes during cell division. Microtubules maintain and determine the shape of the cell. They also play a role in cellular motility. They are composed of alpha and beta tubulin subunits that are assembled into protofilament, ribosomes and peroxisomes. Ribosomes as organelles are granular and small dot-like structures. Ribosomes are concerned with protein synthesis in the cell. They are also called protein factories. Some ribosomes are attached to the endoplasmic reticulum, while others are freely distributed in the cytoplasm. They play a vital role in the synthesis of mRNA. Peroxisomes or microbodies are membrane-limited vesicles like the lysosomes. Unlike lysosomes, peroxisomes are pinched off from the endoplasmic reticulum and not from the Golgi apparatus. Peroxisomes contain some oxidative enzymes such as catalase, urate oxidase, and amino acid oxidase. Its functions include breakdown of excess fatty acids, detoxification of hydrogen peroxide and other metabolic products, oxygen utilization, acceleration of gluconeogenesis, degradation of purine to uric acid, role in the formation of myelin and bile acids. That is all about the basic concepts of physiology that covers the cells and their types, cell membrane and its structural composition, and cell organelles, the working unit of the cell which bring about all the cellular activity. Stay tuned to sqadia.com, where we will be covering other topics of physiology that will help you understand these concepts easily.